All right, well, we'll get started. It's 8 o'clock. Welcome everybody to Grand Rounds. Today is a special Grand Rounds. So Dr. Lee Eckhart is going to be talking to us today. And as you all might know, Dr. Eckhart is the inaugural holder of the Gary and Marie Weiner Professorship in Cardiovascular Medicine Research. And I just wanted to give a big shout out to Gary and Marie. They have been long-term supporters of the Cardiovascular Medicine Division here since for a number of years, and in 2020 they made the commitment to establish this endowed professorship. And I think I can speak for myself and Lee and Mohammed that we're really looking forward to seeing how their generosity will continue to accelerate Lee's research and move her work and the field forward. And I know Gary and Marie aren't able to tune in live, but they are hoping to watch the recording. So Gary and Marie, thank you for everything that you've done to support cardiovascular medicine research, and we are so grateful for your generosity. With that, I will turn it over to Dr. Hamden for the introductions.
Thank you, Dr. Schnapp. Well, it gives me great pleasure to introduce Dr. Lee Eckhart, Associate Professor of Medicine, and Gary and Marie Weiner Professor in Cardiovascular Medicine Research. Dr. Eckhart went to medical school at Georgetown University School of Medicine, where she stayed for her residency in internal medicine. She came to UW Medicine for her fellowship in cardiovascular medicine, and then went to the Brigham and Women's Hospital in Boston for her fellowship in cardiac electrophysiology. In 2008, we were fortunate to recruit her as Assistant Professor of Medicine on the CHS track and co-director of the Inherited Arrhythmia Clinic. In 2012, she transferred to the tenure track, and 5 years later, she was promoted to Associate Professor of Medicine. Dr. Eckhart is a leader in the field of cardiac electrophysiology. Her research is focused on both genetic and acquired causes of cardiac arrhythmias. Her team discovered and characterized cardiac cellular microdomains for the potassium inward rectifier 2.1 with caveolin-3 and the implications for long QT syndrome and ventricular arrhythmias and heart failure. Her team was also the first to create and characterize a transgenic animal model with a KCNJ2 mutation and the other underpinnings for potassium inward rectifier 2.1's role in bidirectional VT and polymorphic VT. Dr. Eckhart has published over 50 articles and book chapters and had continuous NIH funding since 2014, including a recent R1 grant that was just funded titled Deep Mutation Scanning and Function Analysis of Repolarization Determinants. In recognition for her excellence in research, Dr. Eckhart received the Benjamin and Marion Schuster Prize for Research Excellence and the American Journal of Physiology Heart and Circulatory Physiology Best Paper Award in 2017. She has given several national presentations and was recently elected as the Vice President for the Cardiac Electrophysiology Society, which is an international society of scientists and physicians interested in cardiac electrophysiology and arrhythmias. Last August, as Dr. Schnapp mentioned, Dr. Eckhart was named the inaugural Gary and Marie Weiner Professor in Cardiovascular Medicine Research. This is the second major gift from Gary and Marie to the Department of Medicine. And I'm not sure if they were able to join us today or not. Dr. Schnapp alluded to that, but I would like to echo her sentiments and express our deepest gratitude to their generosity. Dr. Eckhart is also a great mentor. She has mentored several pre-doc students who went on to receive research awards, including the Goldwater Scholarship, Hilldale, and Shapiro Awards. In addition, she has mentored several postdocs who currently hold primary cardiovascular research positions. Just recently, one of her postdocs, Dr. Saba Munawar, received an HRS Research Fellowship Award. This is a very competitive award that provides funding for 1 year in addition to the several benefits with the Heart Rhythm Society. In addition to excellence in research and mentoring, Dr. Eckhart is also an outstanding clinician and colleague. She has pioneered fluoroscopy-free catheter ablation and has been training our fellows how to do procedures with less fluoroscopy and at times even no fluoroscopy at all. She's the co-director of the UW Health Inherited Arrhythmia Clinic, as I mentioned earlier, and the Cardiac Arrest Prevention Program. She's the director of the EKG screening program for all UW athletes and spirit squads, and she's been doing this for 5 years. She's a director of the Upper Midwest Southern Cardiac Death Biobank and has received the UW Health Patient Experience Physician Champion Award. Finally, I want to say that Dr. Eckhart is also a great citizen. She serves on several national research and service committees, grant review committees, and editorial boards, including the Journal of American College of Cardiology, Clinical Electrophysiology, and Circulation, Arrhythmia, and Electrophysiology. Today, Dr. Eckhart is going to be talking about her favorite topic, inherited arrhythmias. The title of her presentation is Genetic Arrhythmia Syndrome, a functional genomics approach to define sudden death mechanism. Dr. Eckhart?
Great. Thank you so much for that kind introduction, and also for the opportunity to present some of my research here. My research lab does functional genomics, and we do this mostly identifying various arrhythmia mechanisms related to sudden cardiac death within the cellular and molecular arrhythmia research programs. And here are my disclosures. My goals today really are just to outline the strength of the model that we have in the inherited arrhythmia clinic for not just clinical care for inherited arrhythmias, but how that then inspires research within my lab as well as other labs locally and across the country. You know, first and foremost, we all are doctors and take care of patients. And the inherited arrhythmia clinic is part of a broader scope of cardiac genetics, glued together by Kate Orland, who's a genetic counselor, and made up of both adult cardiologists and pediatric cardiologists. And I want to just give you an idea of the scope of the work that we do in the inherited arrhythmia clinic. These are some newer numbers put together by a research intern in my lab, Molly Melnick, and who's has been doing some cleaning up of the database. And within the inherited arrhythmia clinic, we have over 800 patients that we actively managing, with over 200 unique diagnoses, which go beyond just their inherited arrhythmia syndromes, and including their comorbidities. This has been a source for research, and there have been several induced pluripotent stem cell lines made from patients that we actively manage. And not only in our database do we have the patients, but because we are actively involved in managing them, we have granular data from their defibrillators, clinical management, Holter monitors. And this not only is helpful for research collaboration, but clinical care. And I'm just going to show a few patients to highlight what I mean by that. Again, to give you a scope of how this clinic has changed since I joined the faculty in 2008. As you can see this graph on the left-hand side, we had about 25 patients in the inherited arrhythmia clinic, and now has enjoyed some exponential growth. That growth has also allowed us to engage in international collaboration, such as one that I'm involved in called the Arrhythmia Genetics Network. When you deal with very rare diseases, it is best to be a good collaborator and be able to share with other people who take care of rare diseases around the world. So the Arrhythmia Genetics Network is a group of investigators who all take care of inherited arrhythmias, and we have de-identified patient data to help try to understand some of these syndromes better in a more comprehensive, deep phenotype way. So the structure of the inherited arrhythmia clinic, really, it starts with seeing the patients. We incorporate genetics, and then a lot of this is inspired basic science research. But I'm going to also talk about today the strength of the clinical model for this and how this has impacted patient care, and then talk about how some of the unanswered questions that we have from the clinic has then inspired research. The data I'm going to show related to the research can be a little bit heavy. Sorry about that. Can be a little bit heavy. There'll be, IV curves and discussion of action potentials, and visually it's a little dense, intellectually it's a little bit dense. And so, as my next slide just alluded to, I'll give us some visual mental breaks occasionally with just cute dog pictures to help reset. So to get started, I'd just like to introduce 3 cases that I think highlight some of the special features of the inherited arrhythmia clinic. This is an ECG tracing from a patient of mine who had a cardiac arrest at the age of 19 when he was in the middle of an argument with his cousin. He'd actually had a long-standing history of syncope related to, again, high emotional states, and was told by neurology that he had partial complex seizures and neurocardiogenic syncope, despite the fact that he had always had a normal EEG and didn't have other features that go along with neurocardiogenic syncope. After his arrest, he had a giant workup, including EEG, echo, coronary angiography, toxicology screen, and everything was normal. And so when I put this clinical picture together, I was just asked to put his defibrillator in, but it's always best to try to understand, why it is that this person had this arrest. And phenotypically, we didn't really have much, but we did have a strong history of emotional and stress-induced sudden death. In his case, aborted sudden death. I suspected that he had catecholaminergic polymorphic VT, which we abbreviate CPVT because that's kind of a mouthful to get out with some frequency. Basically, this syndrome is stress-induced death, either exercise, emotional triggers, anything for which there's an adrenergic surge. These individuals can have syncope related to bidirectional ventricular tachycardia, which is the signature arrhythmia for this condition that can degenerate to ventricular fibrillation, as shown in this tracing here from Silvia Priori's 2001 circulation paper. These arrhythmias are dependent on calcium overload and generation of delayed after depolarizations. By definition, they have normal resting ECGs, structurally normal hearts, but it is one of the more lethal inherited arrhythmia syndromes, and approximately 30 to 50% of people are dead by the age of 30 unless these are appropriately recognized and treated. So we suspected that this individual probably harbored a genetic mutation related to CPVT. There are a few genetic arrhythmia syndromes for which you can do genetic testing for them, and it can be not just help supportive, but can be diagnostic. Long QT syndrome, CPVT, and hypertrophic cardiomyopathy are such diagnoses in which the genotype can actually help you support the diagnosis. So back in the day, there were only a handful of genes that we tested that were associated with CPVT, rather bidirectional ventricular tachycardia related to stress. And so we tested him for those in the tiny white print, which you don't really need to read, but it just gives you a laundry list of what we would usually broaden out to test for currently. And not too surprisingly for me, he did harbor a mutation of arginine 420 tryptophan, which of the ryanodine receptor. RYR2 encodes for this giant protein, this ryanodine receptor, which sits on the in the membrane that separates the sarcoplasmic reticulum from the cytosol and helps regulate calcium release for excitation, contraction, coupling in myocytes. There are several hot spots as shown in this little graph on the or schematic cartoon, excuse me, on the bottom left-hand side, that certain regions of this giant protein that seem to be implicated in, if there's a mutation there, it's more likely to be related to CPVT. So I'm telling you this story not because, oh, we were so amazing and we identified CPVT because this, in this patient, but more to kind of highlight that the collaboration we have with pediatric cardiology allowed us to, as I take care of this patient longitudinally, give him appropriate care for CPVT, appropriate medications and lifestyle modifications. But then when he had his two children, who both ended up being gene positive for this same RYR2 mutation, they were started on appropriate therapy as infants. And I have them in checkerboard red instead of solid red because they're genotype positive. It's a highly penetrant disease. Would they not have been treated for this, they likely would have had something by ages 3 and 5. At this point. And this is a mechanism of preventing another sudden death in this family. Second case I'd like to talk about, and this will be a little bit of a longer introduction because there's a lot of really interesting features here. And again, highlight the strength of being able to provide this clinical recognition of what the arrhythmia syndrome is. This patient, I'll say to start off with, she had long QT syndrome, has long QT syndrome too, but when she came to me, she had no diagnosis. She was a student athlete, was starting graduate school, and had presented a few years back with exertional cardiac arrest, which was aborted. She had repeat ECGs that showed she had a prolongation of her QT interval corrected for heart rate at 480 to 499. and consistent exertional related syncope. She got a defibrillator for this. Again, this is before I saw her, and she was not treated with any medication. And she had several tracings of her shocks that she got from her device. Now, in the upper right-hand corner, I don't have a tracing of her longest QT intervals, but this is corrected for heart rate. She's at 477, and that's in large part because she was on a beta blocker called Nadolol, which is standard of what we give long QT and CPVT patients. And as such, her heart rate's quite low with that at 46. But her QT is still prolonged, and she still has that notching in some of her limb leads. More fascinating about this patient, though, is that she had a backpack full of these tracings when we used to print everything out on paper from her ICD. And just to orient you to this feature that I have on the bottom part of the screen, this is one of these tracings that she scrolled out and showed me that she'd been carrying around in her backpack and said, like, can you help me not get shocked from my device? And the top, there are two squiggly lines here. The top one is the local dipole electrogram from the right ventricular lead, the RV tip to ring. And then the Candid coil, sort of a pseudo lead 3 ECG lead, because it's the ICD can then looking to the RV coil. And so it's we use that as a kind of a pseudo ECG. What you can see on her ventricular fibrillation tracing is that she has this very characteristic twisting of the points, like crepe, twisted crepe, and called torsade de pointe. Even more interesting, though, is that she has these typical long shorts initiation before her arrhythmia, which you can see I pointed to with this big red arrow on the marker channel. So, you know, just in our first meeting, without even taking a family history, you know, she has prolongation of her QT interval. She's got exercise-induced sudden death and initiation of torsades with long shorts. So I knew she had long QT syndrome. If there's a question, you can use other types of this risk score. This is an old risk scoring system that we still use, was updated by Leah Crody and Peter Schwartz in 2011. This is the old Schwartz score. Essentially look at different features to try to determine whether or not someone is at risk for high risk, intermediate risk, or low probability for long QT syndrome. These ECG features are of course in the absence of medications that would prolong your QT, or electrolyte abnormalities, or clinical features that would make it more likely that you have torsades or long QT, such as diabetic ketoacidosis, liver disease, et cetera. So in the absence of any other health condition, you look at these different biomarkers, add up your score. And for our patient, she already, before I took her family history, she already had a risk score of 6. Not only is it useful, it's useful to know what the diagnosis is, because then that also helps us with initiating mechanistic-based treatment. So we understand the mechanistic basis of long QT syndrome to be an imbalance of the inward and outward currents, meaning either too much depolarizing current or not enough repolarizing current. And this is manifest on surface ECG by a prolongation of the QT interval. And these individuals are at risk for triggered activity of phase 2 early after depolarizations, shown there in the orange and the orange arrows. And this is what can trigger then torsade de pointe. Understanding which type of long QT syndrome you have can also be useful. The 3 most common and arguably the only 3 long QT syndromes, at least from a clinical phenotype standpoint, are long QT syndromes 1, 2, and 3, related to 2 different potassium channels for 1 and 2, and a sodium channel gain of function for long QT syndrome 3. There are different features that you can then counsel your patient regarding as events for long QT syndrome 1 related to gene mutations in KCNQ1 are related, are more likely to happen with sports, running across the playground, or swimming. Loud noises are often triggers for those individuals with long QT syndrome 2, which is related to mutations in the HERG channel. And then long QT syndrome 3, they have very rare events, but they often occur at nighttime. And so different ways of counseling your patient is helpful to know which type of long QT syndrome you have. But just knowing what type not only knowing what gene is involved, but even where that mutation is. So again, we've got decades of research on long QT syndromes that help us risk stratify patients. What I'm showing here is a cartoon in the upper left of the KCNQ1 channel related to long QT syndrome 1. Turns out that if you have gene mutation in one of those gray boxed regions, which is the transmembrane segments, that those confer a higher risk of arrhythmic event compared to those that are in the C terminus. Doesn't mean you won't have an event, but they're higher risk, particularly in the pore region and in those transmembrane segments. Type of mutation is also useful to know, whether it's missense or nonsense, missense being more lethal than the nonsense mutations. Besides where the mutation is, again, also knowing features that you can anticipate for your patient as they go through their normal life, and you take care of them through their normal life and shepherd them through women going through the process of being pregnant and the delivery and postpartum. So we work closely with maternal fetal medicine group on pregnant patients. And for long QT syndrome, shown in the upper left-hand side, you can see this giant bar, which represents event rates that increase 9 months after delivery. This is almost all dominantly driven by long QT2 patients, as shown on the graph on the right here. This tall checkerboard bar graph indicates the period of time 9 months postpartum, and that is the most vulnerable time for a long QT patient with long QT syndrome 2. Also for long QT syndrome 1, and whether this is related to hormone shifts is most likely. Loud noises are often common for either alarm clocks to wake up to feed the baby, or baby crying maybe can also trigger events. So I show this because this has also helped us with our patient who was undiagnosed. And then we've been able to identify other individuals in her family who were potentially at risk for sudden cardiac death, and then helped her make it through 2 healthy pregnancies and postpartum period. And both of her kids ended up being gene negative, and she was adequately managed during her postpartum period, such that she's able to have those children. So again, another highlight of this specific patient who we identified as having a mutation in her specific mutation, H70R, has been very well characterized by Li Feng and Tim Camp's lab, because we made iPS cardiomyocytes from her, and has led to some really interesting research in that realm. The third case, this will be brief, but again, just to highlight the strength of longitudinally managing patients, as is our model in the inherited arrhythmia clinic, is this case of Brugada syndrome. This individual, who's my patient, the square here, 37-year-old with Brugada syndrome, got a primary prevention ICD related to the fact that he was identified through the fact that his mother had sudden cardiac death in her sleep. She was taken care of by a referring electrophysiologist who had done an atrial flutter ablation on that person when she had atrial flutter in her 30s. So very young, structurally normal heart otherwise, and then died in her sleep. Her daughter also has atrial flutter and conduction system disease. As is often the case, Brugada syndrome ventricular arrhythmia is more likely to occur in middle-aged individuals with an average age of onset, around 35 to 45, and has a male predilection. This ECG I'm showing here is that of my patient who has a spontaneous type 1 ECG pattern. All of the brother and the sister both have a mutation in SCN5A of D356N, which has both Brugada syndrome phenotype in my patient, but then also has conduction system abnormality and atrial flutter, as is typical for these SCN5A mutations. So again, I show you this to kind of highlight the features of being able to proactively identify patients through cascade family screening. And Brugada syndrome is a good one to be able to identify early on, because this is can be a lethal sudden death syndrome, in the sense that These can often be unexpected because they very rarely will have a prodrome, and people can present with sudden death. The ECG shows a very typical, just like my patients, very typical ECG pattern that you'd expect to see in Brugada syndrome. There's a male post-adolescence predominance to the arrhythmia and events. And unfortunately, medications have been classically completely ineffective for Brugada syndrome, and defibrillators are the only proven mechanism to prevent sudden death. Although if you can find someone who can tolerate quinidine, that may end up being fairly useful for them. One of the features I'm going to talk about a little bit later becomes very frustrating with Brugada syndrome, in the sense that you have approximately 30% of patients have a mutation in SCN5A, which sounds like not that bad, but that's 70% of patients don't have a genetic diagnosis. And so it's very difficult to then do cascade family screening or even try to understand the genetics of the syndrome and mechanisms of treatment than if we don't necessarily know what the pathophysiology is from a genetic standpoint. Okay, so those are three stories that I think I hope I've highlighted the benefits of undoing deep clinical phenotyping, genotype assessment for phenotype-directed treatment, cascade family screening, longitudinal family care. And all of those patients kind of fit into some nice diagnostic boxes. And you can do prognostication and offer mechanistic-based treatment for those. The rest of my talk, I'm going to be talking about when that goes awry, and it's more common than you think. And I think that we have a desire to give labels to patients, and I have this, trying to. But if the label doesn't fit, then you're probably doing something wrong, and you may end up hurting the patient. So I have several examples of kind of square peg round hole phenomena in which we've been trying to unravel the arrhythmia mechanism in the research lab. So again, I'll start with a patient. This is a case of a 33-year-old. She had 20 years of exertional dyspnea and syncope, but her resting ECG was normal. She'd been to see lots of physicians, and she'd always kind of blown it off and thought, well, nothing bad has ever happened. I just get really lightheaded. Someone put a Holter monitor on her, and as soon as she starts doing anything, carrying a load of laundry upstairs, chasing the dog in the backyard, she has ventricular bigeminy, polymorphic VT, some things that look more like bidirectional VT, to the tune of, in 48 hours, she has over 1,600 short runs of polymorphic VT, the longest being 22 beats shown here, a lot of it looking like bidirectional VT, despite the fact that she has structurally normal heart and normal resting ECG. So she actually had genetic testing at an outside hospital and was recognized to have a gene mutation in KCNJ2. KCNJ2 is a gene that encodes for the potassium inward rectifier KR2.1, and this is the molecular component that dominantly makes up the current IK1, shown here with my big red arrow down here on the bottom left. This is active in phase 3 repolarization and helps stabilize the resting membrane potential. The current does that. When I was a postdoc in John Mikilski's lab, I'd actually studied her specific mutation, which was in an unrelated person from a different data set. But she'd found this paper online and then came to see us in clinic in relation to some of this work that we'd done. So I had a strong interest in this protein and the specific mutation, but none of it really made sense because these were so-called long QT syndromes, and specifically called long QT syndrome 7. And this is a list of long QT syndrome risks genes. And hers was considered, in KCNJ2 is considered, loss of functions are considered part of long QT7, but never really made that much sense to me because they don't have long QT and they don't have torsades and they have bidirectional VT more like CPVT. So what is the clinical phenotype and what, what is the arrhythmia mechanism? Well, to sort that out, we decided to make a mouse out of this specific patient. The mouse work, I'll just highlight, is done by Dr. Louise Riley, picture shown on the bottom right. And what Dr. Riley showed is that the mice, just like the human counterpart, structurally normal heart, normal resting ECG, normal physiology otherwise until they're under stress. And then the mice have bidirectional VT, just like the patient did. She looked at this further to sort out what was happening with the ionic currents. I'm not going to go through all of this in detail, but I do just want to highlight a couple features. We studied this mutation in a couple of different ways. One was work done by Matt Kalscher when he was a postdoc in my lab, and he measured currents in heterologous expression using the specific mutation with wild type. And then Dr. Riley did this electrophysiology characterization in the mouse. What I want you to see is this colorful figure in the center left, which is that in normal physiology, the wild type KR 2.1 current under stressful conditions augments its current for more repolarization drive and stabilization of the resting membrane potential under adrenergic stress. But this mutation confers loss of function, as you can see by this bar graph, that these ventricular myocytes from the mouse then have a loss of function. This then manifests by the action potential of these mice as a longer action potential. So less repolarization drive, longer QT interval, longer action potentials for the mice. Seems like we're kind of looking at a long QT type phenotype after all. But they have bidirectional ventricular tachycardia. And so the association with catecholinergic polymorphic VT and calcium mishandling in that syndrome, can't be ignored. So, and this goes beyond just semantics, okay, because the way you approach the initiation of arrhythmia or prevention of initiation for arrhythmia for these two different syndromes, long QT syndrome versus CPVT, is divergent. If you have long QT syndrome and a phase 2 EAD trigger, you treat those patients with atrial pacing, avoid bradycardia, give them avoidance of QT prolonging drugs, electrolyte supplementation for membrane stabilization, and ICDs if they have syncope and sudden death despite all the above. But DAD triggers, then you avoid tachycardia. There's more of a role for flecainide for refractory arrhythmia and left cardiac sympathectomy in refractory cases, and less of a role for ICDs because of the perpetuation of calcium overload that can occur with ICDs. So to get to the bottom of what the arrhythmia trigger was as an important one, Dr. Riley partnered with Francisco Alvarado to look at the calcium handling and to sort out why are they having this bidirectional ventricular tachycardia. Maybe they have a lot of delayed after depolarizations related to calcium mishandling. And actually, they found out that it was completely normal, to our surprise, that the calcium handling, the wild type and the R67Q looked identical. But what wasn't identical was the predilection for the onset of these funny looking early after depolarizations. And I say they're kind of funny looking because I kept pressing Dr. Riley, aren't you seeing delayed after depolarizations after pacing rapidly? And in the center here, you see these small membrane oscillations, which are technically delayed after depolarizations, but none of them really reached threshold. What was much more common though, in the R67Q mice only, and not at all in the wild type, are these early after depolarizations shown at the top and with my big blue arrow here. These are unusual and different than phase 2 EADs in the sense that their time from the preceding action potential stimulus is much tighter coupling interval, and their takeoff potential is much more negative. These are so-called phase 3 EADs. I suppose we shouldn't have been too surprised that we saw phase 3 EADs in the setting of loss of function of IK1, which is the active repolarizing current during phase 3 of repolarization. Okay, so that was a lot of talk about EADs and DADs and action potentials. And so we'll just take a short little puppy break before I really talk about the reason I'm showing you this, which is, again, to sort out how we're going to optimize treatment approaches for these patients, because they don't fall into a category of CPVT or long QT syndrome. They are unique. And so in Dr. Alexei Glukoff's lab, we've partnered with Dr. Glukoff and Dr. Lang to do whole heart optical mapping. And we're using this to not just describe what the arrhythmia mechanism is, but to optimize treatment approaches for these patients. And while we've been doing all this work at the basic science level, a lot of the individuals who had been busy publishing long QT susceptibility genes, as listed here, these 17 different genes, took a kind of a second look, and there's been a big reappraisal of genes reported to cause long QT syndrome in this publication that's now 2 years old. And putting the level of evidence of genetic as well as experimental evidence, a lot of these long QT susceptibility genes are, maybe perhaps not long QT syndrome, which we also then figured out. And I think it's important to take a second look and do redefinition of these syndromes, because mislabeling, like these bags of corn labeled onions, you end up with the wrong product and you end up treating the patient the wrong way. So while the list of long QT syndromes and their associated genes are listed here, this might change in years to come to be a little bit more refined to something a bit shorter, but not just yet. Okay, changing gears a little bit and to talk about genetic variant classification. A giant conundrum and problem for functional genomics in this day and age is trying to keep pace with the number of genetic variants that have been identified. If you consider how the speed, accuracy, and depth of sequencing has exponentially grown, really kind of about starting about 15 years ago, from in the 1980s when I was doing these manual slab gels, the field of functional genomics could keep pace with this because about the same frequency as you would identify a genetic variant, then there could be functional studies to keep pace with that. But now we have so many more genetic variants identified than are specifically classified. To highlight this, I'll turn to the KR2.1 protein again, in which this is a pretty small protein with just over 400 amino acids. But the majority of those variants that are published in ClinVar are variants of undetermined significance. And this is a problem because while I highlighted the fact that genetic testing can be diagnostic in some settings, and can be supportive of other diagnoses, if you have a variant of unknown significance, it's not a clinically actionable entity. And so, it might be true that they have that variant, but it may not be related to that disease. And so again, this is open to a lot of misinterpretation and mislabeling. And so trying to get to a place where we can keep pace with these identification of genetic variants is something that I've been interested in, and as well has been something that Dr. Corey Anderson, who's a scientist in my lab, has been working on for literally for decades. He did a PhD in Dr. Craig January's lab, at which time he was working on hERG mutations. I won't go through all of this in detail, but I just want to point out a few features of this so that what I'm saying makes sense. One way of looking at ion channels is to measure their current and determine and you can determine pathophysiology that way, but this is time and labor intensive. The majority of long QT2 related hERG mutations are pathogenic because the proteins are misfolded and they don't reach the surface membrane. Some of these can be rescued through temperature as well as medications because they're not quite as dysfunctional. And so in this bottom figure here, the degree of misfolding is likely related to the degree of ability or inability to rescue that from that trafficking deficiency. Dr. Anderson worked on this by doing a series of Western blots, which can identify these trafficking deficient mutations. But Western blots, as anyone who's done them knows, can be a little fickle. So he then, again, predicated on the concept that a misfolded protein it would be unstable and then would form disordered aggregates inside the cell. He used a E. coli based solubility assay to look at all of these different long QT2 related gene mutations and assess their solubility. And what I'm showing you here, this large figure on the bottom, this bar graph, you can see that the degree of protein misfolding correlates correlates with the relative solubility, and then is parallel to what you see with respect to the trafficking phenotype. So trafficking phenotype in the sense that those that could not be rescued, so very misfolded, very dysfunctional proteins are shown in red, and then yellow are less disordered, and the blue are even more, and then the dark blue are more like wild type. So the solubility assay for these specific domains, the hERG channel, the wild type have normal solubility, and the more dysfunctional the protein, then the more insoluble it is. Dr. Anderson then took this a step further, looking at other proteins, again with the idea that misfolded proteins will be unstable and form cellular aggregates. This was directed at laminin variants. Laminin is a structural protein that's associated with a very aggressive type of cardiomyopathy, as well as skeletal myopathies, lipodystrophy, and premature aging. And what he found by employing this solubility assay and also looking for cellular aggregates, again, these markers of protein instability, is that he could classify nearly all of the skeletal and cardiac muscle pathogenic mutations in laminin could be categorized by employing these high throughput assays. So you can't make, all of these hundreds of mice from all these different variants, but you can employ these more higher throughput assays in order to at least understand where on the spectrum these variants potentially are. So the laminin data, you can just focus on the graph on the right side, in which you can see increased cellular aggregates also associated with decreased solubility. And so the pattern fit, again, mostly for skeletal and cardiomyopathic laminin variants. So this is the type of thing that can be used then to employ for more proactive approach for identifying variants, instead of always trying to keep pace with the identified variants in that are being found in the clinic. What we have we literally just got funded to do this work, and to look at, and in this work we'll be doing what's called a deep mutational scan for KR 2.1, in which we will, using next generation sequencing, engineer all the possible genetic variants that could occur in this protein. And then, and we obviously can't, make a mouse out of all of those different variants, or even iPS cells out of all those variants, but instead we'll plan to do these high throughput assays on a massive parallel scale to sort out where on the spectrum of normal, abnormal. So in the center here, I'm showing this yeast assay, which is a really, it's a really lovely system in the sense that these yeast are engineered to be deficient in potassium channel. And if you grow, if you grow them on high potassium agar, if you engineer a wild type channel into that yeast, then the cell can't handle the potassium inside the cell. There's no pump to extrude the potassium. It becomes toxic, and then the cells die. So wild type will grow, but then if you expose it to potassium, then the wild type potassium channel then becomes a toxic feature, and the yeast die. So if you have a protein, if you have a channel mutation and that protein is dysfunctional for any reason, trafficking, gating, or PIP2 binding, no matter what the abnormality is, the yeast assay can pick that up, because it will, those channels will grow because it doesn't pass current. Dr. Saba Manwar will also be participating in the study by measuring currents kind of in the traditional fashion in order to highlight the strength of the model that we're doing. We're combining this again at a massive scale with these deep mutational scanning. And then with our colleagues at Mass General and the Broad Institute, we'll mine to the databases of TopMed and the UK biobanks to correlate whether or not any of these repolarization abnormalities that they pick up at the clinical end from these databases then are also correlate with what we're finding with our deep mutational scan for CARE 2.1. And we'll also plan to look at other potassium channels as well. So this is evolving work. You'll see more of this hopefully sometime in the future. Okay, short little puppy break here. Moving on to just a clinical project that we've started again in collaboration with folks around the country, and that is to help uncover some of the unknown genomic features in Brugada syndrome. I presented one case of Brugada syndrome in which there's a clear pathogenic SCN5A mutation, but as I said, about 70% of individuals like this individual shown here, whose ECG is shown here, who's had 2 episodes of clear cardiac related syncope, atrial flutter at age 11 on a hot summer day, and is found to have a genetic variant classified as benign pseudodeficiency, which may be not specifically pathogenic, but it may alter channel expression and very difficult to ascribe specific pathology. There's a lot in the way of genetics for Brugada syndrome that we don't understand, as 70% of people don't have identified specific variants. And it's probably because this disorder is not really monogenic or follow a Mendelian inheritance pattern like long QT syndrome and CPVT, but more likely to follow oligogenic inheritance pattern where there are several different rare variants that combine together that create this pathology. And so to get to the bottom of this, again, with rare diseases, it can be very challenging. And so we designed this project. It's called Link My Heart. And you can Google Link My Heart or Brugada project or brugadalinkmyheart.org. And together with the Broad Institute, MGH, Stanford, and Vanderbilt, and UW, we have created this database that is inspired by anti-siloed research. What I mean by that is when you take care of individuals with rare diseases, if we broaden our capture of patients, we'll have a broader understanding. And so everyone has these silos of databases. And so instead of even just trying to combine silos, we thought that we would offer something direct to patients who tend to be the most motivated to find answers for their disease. So this is a direct to patient. They enroll at their own will, provide their ECG data and any other medical data, and then we send them a spit kit. The spit kit then comes back to the Broad Institute. We'll do whole genome sequencing, and again, to capture a more broad, complex genomics around Brugada syndrome. Also for UW, this really presents a unique capacity potentially for additional functional studies to create iPS cells from a much more broad phenotypic capture. Okay, in the last 5 minutes, I want to introduce a topic that is evolving. I don't have answers for this yet, but I've got some ideas of how we should approach one of the most difficult and pressing issues in the inherited arrhythmia clinic. Again, it's a story of square peg round hole with undefined arrhythmia syndromes. We'll start at the clinic level with a patient example. This is a 27-year-old woman who was previously completely healthy, not taking any medications, who had a cardiac arrest while driving in stressful traffic. She had a prodrome of, I'm gonna pass out, and then proceeded to have ventricular fibrillation. All of these tracings shown here are from her AED. She had over 7 shocks before she was then able to have return of spontaneous circulation. She luckily survived, made a full neurologic recovery, likely related to her age, and underwent deep phenotyping. And you'll probably guess that the phenotyping really didn't show anything. She had a negative tox screen. Her CT angio was normal. Her ECGs, resting ECGs were normal. Her cardiac MRI was normal. It was normal. Even her treadmill. So then diagnostically, you might be thinking, oh, maybe she's got CPVT like that first patient that, that you talked about. And so we did a broad genetic panel, and she's got a bag of four different variants of undetermined significance, all of which these genes are associated with different genetic arrhythmia syndromes. So whatever she has might be related to those, but it might not. And this becomes really problematic when you start talking about trying to employ treatment. The dirty little secret of a lot of inherited arrhythmia clinics around the world, and including ours, and including the largest such published database, which is the Casper Registry out of Canada, is that, well, a lot of patients fit into these neat diagnostic boxes of genotype-phenotype correlations, and I've shown the strength of being able to understand that. About half of persons don't have those clinical phenotypes. And so, and a lot of those individuals harbor uncommon genetic variants of undetermined significance. So modeling these individuals and trying to figure out why is it that this person with a completely normal heart just died, and how am I going to approach them, becomes it's a big problem, because there's no mechanistic treatment path. We have no methods for prognosis, cascade family screening, and then all the treatments are trial and error. I mean, educated trial and error, but they're trial and error nonetheless. So, we thought that perhaps we could, take a step away from this classic path of you define a clinical phenotype, you have a syndrome diagnosis, you make a genetic diagnosis, and then you have syndrome-specific treatment. For these undefined phenotypes, we don't have those answers, but what we do have is the capability of taking creative approaches to change the way that we would normally look at the physiology of this syndrome to try to understand it better. So it's my idea to then create patient-specific iPS cardiomyocytes and then perform multi-scale analysis, functional characteristics of these iPS cardiomyocytes, as well as multi-omics, genomics, transcriptomics, and proteomics, in order to try to start to unravel what is the underlying mechanistic phenotype for these idiopathic ventricular fibrillation patients. Because you can't model them in any way, because you don't know where to start with the model. You have no biomarkers to go with. Even if you created a mouse with all those mutations, you wouldn't know whether or not would be the same clinical phenotype as what the patient has. So all we have is the capability of then going to the stem cell level. So we have started some of this work. These are some single cell optical mapping traces recorded by a student in my lab who is graduating this year and going on to medical school at UW-Madison, Manasa Kalori. Did single cell optical mapping of an idiopathic ventricular fibrillation patient. And we're using this to then characterize electrically what these people have. The concept, it's not what we've done here. This is just an example of how this could potentially work, is that then we will take these constructs and then employ antiarrhythmic drugs on top of those. And so that we're experimenting in the dish instead of on the patients with our trial and error antiarrhythmic drug approach. Eventually, we will get to, hopefully in the next 6 to 12 months, we'll then be starting to do not just single cell optical mapping, but then optical mapping on these constructs that my lab has been a more peripheral part of, but has really come out of Wendy Cron's lab and her former PhD student, Britt Napawaki, in which PDMS stamps pattern constructs allow the cardiomyocytes to align their myofibrils in end-to-end myocytes so that it's a more physiologic platform for the cells to grow. And then also in collaboration with the Tim Camp's lab and Junhua Zhang, we recently published this work again with our collaborator Wendy Cron and Brett Napawaki and Alana Stempian looking at the optical mapping, which was done in Alexei Glukov's labs. You can see there's a lot of collaboration. The benefit of not just culturing the cardiomyocytes by themselves, but then also with patient-specific cardiac fibroblasts. So the idea is that we will have patient-specific cardiomyocytes and cardiac fibroblasts, because the cardiac fibroblasts co-cultured with the cardiomyocytes allow for, again, more physiologic substrate, and then we've shown that this increases calcium rise time as well as contractile strain. And so electrical features that will hopefully be able to make the most physiologic type platform that we can come up with in order to compare these. And again, this is evolving, but we're really hoping that we'll get this off the ground and combine this then with these functional characterizations with the multi-omic approach, again, with the idea that we'd like to get to the bottom of these mechanisms in order to be able to treat our patients more effectively. Small puppy break before I just bring a summary to the work that I've described. The model for the inherited arrhythmia clinic is really a collaboration of basic science and clinical EP with cardiac genetics. And I hope I've been able to show the benefits of understanding genotype-phenotype relationships, but then how you can employ basic science methods to find solutions for undefined features, not only arrhythmia mechanisms, but toward variant characterization, genomics, and then some innovative approaches for undefined arrhythmia syndromes. I'd really like to thank all the amazing people in my lab that I get to work with. Shown here in the big picture on the top are my students, research interns, Doctors Riley, Anderson, and Munwar, and as well as all my fabulous colleagues in the cellular molecular arrhythmia research program, many of whom I've called out specific collaborations with the work that I presented here, including Tim Campbell, Alexei Glukov, and Francisco Alvarado. And as well as collaborators in the CVRC, the Go Lab, Crone Lab, and then my collaborators in the CVRC T32, and then of course the Inherited Arrhythmia Clinic. I'd also like to give a shout out to my funding agencies and the Gary Marie Weiner Professor of Cardiovascular Medicine Research that make all of this actually possible to do. Thank you so much for your attention. And I think we have a few minutes for questions.
Dr. Eckhart, thank you so much for the excellent talk. The team science is amazing, and congratulations again on your R01 grant. So we have a few minutes for questions, and let me start with the question, Dr. Eckhart. So you see a patient in clinic, 25-year-old, and they have a QT corrected of 485 milliseconds. Mm-hmm. They're taking a medication that is known to prolong QT, but they need that medication long term.
Yep.
Patient is asymptomatic,
mm-hmm.
and that EKG is otherwise unremarkable except for the QTC. Mm-hmm. What would you recommend for this patient?
So that's a good question, and not an easy one. And actually, I got together with a couple other long QT experts a few years ago. We wrote this paper around these messy clinical things that inevitably, again, there's no specific answer. But I think that a lot of people take QT prolonging medication, but not everyone has manifest QT prolongation. So I think the first thing I do is actually look to see whether or not that medication uncovered a long QT susceptibility. And we do genotyping to see whether or not they have a long QT susceptibility genetic variant to begin with, family history, and then to see if that's if that's part of the entity. You can give them beta blockers if they can take it to try to treat them as though they have long QT syndrome, and then just see if they have symptoms. If there's excessive prolongation, like over 500 milliseconds, there might be a potential role for, if there's no way they can get off that medication, and you've given them electrolyte supplementation until there's untoward things happening at the other end of their body with all the magnesium that you there may be a role for more aggressive therapy, but for sure try to get to, is there another underlying etiology for that first, and then see what you can do with electrolyte supplementation, beta blockade to keep them otherwise asymptomatic. And if they don't have symptoms from that, then it's more just don't take anything else that's QT prolonging. And, you know, that's, and get your thyroid checked, you know, because that situation plus thyroid storm then would equal, you know, risk for sudden death.
Thank you. As you know, that's a common scenario.
It sure is. Yeah.
I have a couple of questions. So wonderful talk. Love the team science approach to it all. It's truly bench to bedside, back to bench and back to bedside. So can you just talk or touch upon the future of gene therapy for correcting some of these known genetic mutations in patients?
Yes. You know, in the abstract sense, in the sense that I think that, you know, I didn't talk about this because it's a beautiful talk in and of itself, to talk about what we can do, not just diagnostically and prognostically with genetic testing, but understanding if it really is truly a single genetic variant on a single allele that a person has that's causing these potentially fatal conditions, of whether or not you can correct that is probably becoming more attainable in the sense that CRISPR prime editing is being done in iPS cells as well as in mice right now. And maybe it's something that will be useful for patients in the future. It's a way of doing single strand DNA cuts instead of the double stranded cuts with the prime editing. And it's more likely to be useful for these types of genetic disorders. And as you know, it's already being used for sickle cell anemia on patients. But in that sense, you can take the organ out of the body and then do what you do and then put it back in. And so for the heart, it's likely to be a little bit more complicated, but you know, I'm hoping I'm going to be out of a job soon. So.
Yeah. And then my second question is, are you using patient-derived iPS cells to determine best medication, best therapeutics for individual patients?
Yes and no. You know that we use iPS cardiomyocytes for disease modeling, and then taking the step to decide, you know, what is mechanistically is going to work best for treatment. That is something that I would like to do. Right now, I'm using my animal models because I need the whole heart to go out of rhythm. I can't make a cell have torsades, and so you know, but we're, like I alluded to, we're putting these constructs together to see if we can engineer arrhythmias in a dish and then try therapeutics on them in that way. But for sure, it's such a powerful tool, and I'm thankful I get to work in this institution where we have labs like Tim Camp's lab that really are at the forefront of creating not just cardiomyocytes, but regional specific ones, and will be a useful research direction for us going forward.
Great. And can I make a shout out to Alexei, who just got tenured, just got promoted.
Yay!
Yay. Strong work. Okay.
All right, well, I think we're past the hour, so thank you again for the wonderful talk, and thank you all for joining, and have a great day, everyone. Bye-bye.
Bye now.